to inform the brain models. Here you see how we use magnetic resonance imaging data to construct brain network models. We parcellate the brain in different brain regions, and each brain region is then represented by a neural field model, represented by mathematical equations. They are interacting through the reconstructed structural connectome, and then we generate different kinds of activities, neuronal activities, but these can be then also translated in fMRI signals via forward models and EEG and MEG signals, for example. We can also then probe interventions. Here you see, for example, an electrode that is inserted in the virtual brain twin. I apologize. So we are not creating maps, multi-scale maps of the brain that are static, but we really can simulate based on the digital avatars, the activity that is generated in the brain. The brain twins are falling under AI, artificial intelligence definition, according to the AI Act that is in force since August this year in Europe. The AI Act, the European Artificial Intelligence Act, is the first regulation in the world for trustworthy AI. And it came in force on August 2nd this year. It has now a grace period, but for many AI systems, it will be applicable in the year 2026. So let us have a look at the definition of AI according to the European AI Act. An AI system is a machine-based system that for explicit or implicit objectives infers from the input it receives how to generate outputs such as predictions, content, recommendations, or decisions that can influence physical or environments, physical or virtual environments. So it is machine-based, that infers from the input it receives how to generate outputs, and it can influence physical or virtual environments. And this is exactly applicable to our human digital brain twins. We are building the brain twins to use them for medical applications, for example, to plan neurosurgical interventions, pharmacological interventions, or stimulation protocols for individual patients. And this is the risk system of the new AI Act in Europe. It differentiates 4 categories of risks: the unacceptable risk, the high risk, transparency risk, and the minimal or no risk. And all medical devices fall, per definition, under high risk if they contain an AI system. And with this, for such medical devices, if they enter the market, they need to undergo ex ante, before entering the market, conformity assessment for trustworthy AI. So how is now trustworthiness in the AI Act defined? Of course, it is relatively difficult to define trustworthiness, but in the AI Act, different categories that contribute to trustworthiness have been outlined: human agency and oversight, technical robustness and safety, privacy and data governance, transparency, diversity, non-discrimination and fairness, societal and environmental well-being, and accountability. So in order to use our digital brain twins for clinical applications, we have to demonstrate and get certified for these different aspects of the AI that is part of our system. In Europe, you might know, we had for 10 years the Human Brain Project funded by the European Commission, and the output, or one output of the European Human Brain Project, was a digital platform for neuroscience, which continues to exist and which is funded by various contributing projects now. It is coordinated by the eBrains AISBL, located in Brussels. And what you see here are core elements of the digital infrastructure. The infrastructure is not centralized. It is federated, and several compute infrastructures are integrated and interoperable to contribute to this European digital neuroscience infrastructure, eBrains. Some core elements are supercomputing centers that provide high-performance computing and storage, but also a central knowledge graph, which provides an interface to query not only data, but also tools, software, and computational models, for example. There are many other tools, for example, cloud environments, where the users who have registered for free at eBrains can create workspaces, either to work privately or to invite other collaborators and work collaboratively in projects. There are several standardized APIs, interfaces, and software developers, research software developers can interface their software products with the core infrastructure elements of eBrains. And we have interfaced different components of the virtual brain software to this infrastructure to make it directly and easily accessible to everyone. So this is described in this publication in case you want to see the details, but I give you a couple of examples. So the core software, the virtual brain, that you can find also under the URL, thevirtualbrain.org, can be directly accessed on eBrains. So this is a graphical user interface. Here you see the URL. Excuse me. It can be run in the cloud, so you do not need to download the software. Of course, you can do this. It's free, it's available, but you can also directly start it in the available cloud resources provided by eBrains, and there you can run simulations. Of course, you can also use the command line interface if you are not in favor of the graphical user interface. In order to generate the inputs for such a brain network model, typically you need to do a lot of processing of the underlying imaging data. And we have created such processing workflows and made them available also via eBrains. So if you have your own data, you can just use these containerized workflows. They run a system independent to then use raw imaging data of different modalities to reconstruct the fiber tracts, generate the structural connectome, parcellate the brain in different brain regions, determine the region centers, and also use the pipelines, for example, to compute forward models for EEG or MEG simulations. With these pipelines, all the processing and analysis becomes fully reproducible. And this contributes, of course, to, on the one hand, the reproducible science, but on the other hand, also the requirements of the European AI Act to make all the algorithms used transparent and robust and reproducible. It also contributes to the possibility to process a large imaging cohort and generate a large number of individualized digital brain trends or brain network models. We have several such workflows developed, not only for healthy adult brains, but here's an example, for example, for lesion brains with stroke lesions, and these pipelines then are adapted to take into account the requirements of such lesioned brains. They also comprise many quality assurance metrics that are generated and can be used to control the meaningfulness and correctness of the processing results. Another example of how eBrains integrates different research software components is the integration between the digital brain atlas coming from the Forschungszentrum Research Center Jülich. They have created, based on microscopic analysis of postmortem human brains, atlases that contain different kinds of information, Sorry, here you see the receptor distributions, for example, and through the API, the Python interface that they are providing, we can use this additional information that comes from the post-mortem brains and integrate it in our brain network models to make the brain network models even more biologically plausible. So these the receptor distributions, for example, can influence or constrain the parameter values of the individual brain regions in such a model. In the recent years, we also have developed an interface for multi-scale co-simulation, where the virtual brain that typically simulates the brain at a pretty coarse level can be interfaced with microscopically detailed neural network simulation frameworks such as NEST or Anarchy. And with this, we can build multi-scale models where some parts of the brain are simulated via spiking neuron models. Here you see such a network, and others via mean field models, and they're interacting and influencing each other via the existing interfaces. So these little balls here are inhibitory and excitatory neurons that are embedded in a full brain model. And this is, by the way, my own brain. This is a reconstruction of my own brain and the multi-scale model of my brain. I want to give you now two examples how we use the digital brain twins for understanding mechanisms, how the brain works. They both are pretty recent, have been published this year and last year. And we start with the study that has been published this year about how the human connectome generates traveling waves in the human brain. Why is it important to understand the mechanisms of traveling waves? Because, as you can see here, the wave fronts are the sites in the brain where we have synchronization, where the activity state across different brain regions is identical. And you might have heard what fires together wires together, so generating such wave fronts at different scales and different speeds, and coordinating the trajectories of such wavefronts is important to orchestrate synchronization and desynchronization in the brain. So we wanted to understand better via computational modeling how these traveling waves are being generated. So let's have another look on the traveling waves. Here you see an example of cortical traveling waves in the marmoset monkey in the area MT. And as you can see here, this is a very small section of the brain. The edge length is only 2 millimeters. And here you see a traveling waves going from the top left corner to the bottom right corner. And we also see the speed of the traveling waves with 0.1 to 0.6 meters per second. This is relatively slow, indicating that intracortical non-myelinated fibers are underlying these traveling waves in area MT of the macaque monkey. So these are local field potentials in the monkey. Now we have another example here of cortical traveling waves in the human brain. Here we see an electrocorticogram, patients with epilepsy, and we see this traveling waves that are circulating counterclockwise around this center. Here the speed of the traveling waves is 2 to 5 meters per second, so it's a little bit faster than the traveling waves on the last slide, indicating that the underlying neurons are myelinated neurons with a higher conduction velocity. These traveling waves can be found everywhere in the brain. So they have pretty well characterized for memory tasks, for example, in different regions in the brain, including in the hippocampus. In the visual system, as we have seen, for example, in area MT of the macaque. than in the motor system, but in principle in all other brain systems, they have been characterized at different temporal and spatial scales. So others have already investigated the generative mechanism of traveling waves, and a first study looked as phase-locked weakly coupled oscillators. And with such phase-locked weakly coupled oscillators, it is certainly possible to generate traveling waves that either travel from one direction to the other or from the other way around. So the question is now, what generates the direction, or what determines the direction of the traveling waves? And here another study has investigated this. They looked at the intrinsic or isolated frequency of such oscillators and the phase shift of the traveling wave. And it has been found that from faster frequencies to slower frequencies, there's this phase shift. So the traveling wave would travel from the faster intrinsic frequency to the lower intrinsic frequency. Another team has now looked at the determinants of the intrinsic frequencies. So here you see the frequencies, and here you see the in-strengths of a network of coupled oscillators. And it has been shown that with increasing in-strengths, so the more connection a node has, the slower is the intrinsic frequency of this oscillator. And we took these results together, and we were hypothesizing that in the brain, the in-strength gradients are generating frequency gradients, as has been shown here in this network of coupled oscillators, and that the frequency gradients then direct the traveling waves, as can be seen here. And we wanted in the study test whether this is the case. And we started very simple. We built a network of coupled oscillators. So this is the work of a PhD student in my lab, Dominic Koller, who just actually is finishing his PhD. And he generated this network of coupled oscillators. And you can see there's an in-strength gradient implemented in this network. The in-strength is indicated here by this color code from low in-strength to high in-strength. And when we now simulate such a network or grid of coupled oscillators with an in-strength gradient, we see that this grid generates clearly traveling waves. If we now instead replace this in-strength gradient with uniform in-strengths, then we see there are also traveling waves, but they are going in all kinds of directions. They are not directed. And this can be also analyzed. And we see now that with the in-strength gradient shown in yellow, we have a very clear directedness of the traveling waves. And if the in-strength gradient is removed and the in-strength is uniform, the traveling waves go in all kinds of directions. So now the next question we wanted to look at is, do we have in-strength gradients in the human brain? And for this, we used the data from the Human Connectome project. We did Scheffer parcellation, and parcellated the brain 1,000 regions and analyzed the in-strength gradient. As you can see here, indeed, there is a gradient in temporal and fronto-orbital and parietal regions. There are lower in-strengths, and in frontal and occipital regions, we find a higher in-strength, and this generates in-strength gradients in the human brain. So now our next question was whether these in-strength gradients that we found in the human connectome would generate traveling waves, as we had hypothesized. And we hypothesized, according to the knowledge we had at this time, that these traveling waves would travel from the regions with low in-strength to the regions with high in-strength. So from the temporal and parietal and fronto-orbital regions to frontal regions and to occipital regions. This was a brain network model that we have first generated. It's a brain network model of coupled oscillators, Kuramoto oscillators. We also take into account the time delays between the brain regions, and we run a simulation, and indeed we see that cortical traveling waves are being generated in such a brain network model. And we wanted to also characterize the direction of these cortical traveling waves. So for this purpose, we computed flow potentials. And here you see now the origins, the sources, and the sinks. Sources are red, and the sinks are shown in blue. And this indicates where the traveling waves are starting and where they are going from the red areas to the blue areas. And this corresponds to the in-strengths that we have previously determined in the human connectomes. So now, as a next step, we again constructed brain network models, but instead of using simple oscillators and the nodes, we replaced the simple oscillators with more biologically grounded physiological brain network circuits consisting of pyramidal neurons, excitatory interneurons, and inhibitory interneurons. The pyramidal neurons received input from the connected brain network and fed activity to the inhibitory and excitatory interneurons. And there were then also feedback connections from the inhibitory and excitatory neurons to the pyramidal cells. And when now constructing such a brain network model, we again simulated this. We found again traveling waves. We analyzed all the time points and all the simulations, and we found that in 90% of all time points that these traveling waves have been generated, and in 80% they followed the in-strength gradient of the human structural connectome. So here you see now the experimental data from patients with epilepsy, so intracortical recordings. The color indicates the frequency of the potential recorded intracranially in these patients. The arrows show the direction of the traveling waves that were also experimentally measured in these 77 patients. And we can see that there's a frequency gradient as well. So the higher frequencies are in the temporal region, and the lower frequencies can be found in the poles on the frontal and also the occipital pole regions. And we see also in terms of the direction that the traveling waves in these measurements go mainly from the temporal regions to the frontal regions and to the occipital regions. Now we go back to our simulations, and we look at the frequency gradients in our simulations. We see that higher frequencies can be found in temporal regions and lower frequencies in frontal and occipital regions. So our simulations are in alignment with these empirical measurements in 77 patients. So taken together, we are able to construct brain network models. In this case, we did it for 776 individual healthy human adult brains. And we were able to understand how the human structural connectome generates these wave patterns and directs the wave fronts through the cortical surface. So now, of course, the question is, how is that influencing cognition? And in the next study that I want to show you today, we looked not only at the dynamics of the brain as generated by digital brain twins, but we also looked at the cognitive performance. So for this purpose, we used also human connectome project data from the US, and we constructed 650 virtual brain twins, as you can see here, and we plugged in these individualized brain network models, circuit models, as you can see here, in the frontal and the postoparietal regions that then were tested for their performance in the 650 individual brain twins. So this is just another depiction of the same thing that I have shown on the previous slide. So this is a brain. Here you see the prefrontal cortex and the postparietal cortex. So this is a circuit model that we used and that we plugged in the brain network model. These two populations were sitting in the prefrontal cortex, and these two populations were sitting in the postparietal cortex. And as you can see, the regions are mutually inhibiting. So there's one pair or one team of populations called A and one team of populations called B. And the A team is inhibiting the B team, and the B team is inhibiting the A team. Within a team, the populations are self-exciting. So the postparietal cortex region A is exciting the prefrontal cortex region A, and the same is the case for the populations B. And now we can feed in evidence in these postparietal regions A and B, and they are competing, because they are mutually inhibiting. If we now choose a very small contrast of evidence or stimulation of the two populations within a noisy network level, it will be difficult for the circuit model to come to the right decision, which of the two populations received is a stronger input. So we simulate this by plugging such a circuit model in each of the 650 individual brain network models. And this is just an example simulation showing the activity time courses of these four guys, colored according to the color here. And we see in this example simulation, this is the time axis, this is the synaptic activity of each of the four populations after 0.5 seconds. The blue populations gain high activity and they reach the high attractor state, the point of no return when a decision is being made. So in this case, the decision is made for A, and this would be a correct decision, because indeed the evidence was higher for population A than the evidence for population B. Excuse me. So now we do another simulation. And here in this example, you see that the green populations are going to the high activity level, the high attractor state of no return. So in this example, despite the evidence being higher for population A than for population B, the green population would win, and this would be an incorrect decision. And this can be done many, many times. We did it 1,000 times per subject, and we measured the ratio of the correct decisions and the incorrect decisions of these simulations. So in this case, there were 790 correct decisions and 210 incorrect decisions. This was done for all 650 individual brain twins. And the results were then compared to the actual performance of the individuals. And we found that the simulation performance that is shown here was correlated significantly with the fluid intelligence of the true individuals behind the digital twins. The fluid intelligence is a capability of deep thinking and difficult problem solving. What we also found was that the decision-making time was positively correlated with the fluid intelligence of the people behind the digital twins. And this was surprising for us because this means that those who score better in a fluid intelligence task, so those that are labeled better thinkers by this test, they take a little bit longer for taking a decision. And we didn't have this data from the empirical study, so we were contacting the Human Connectome Project and asking for this data. And we received the data, and indeed, as we will see in a moment, the data confirmed our model predictions. But let's have a quick look at the test that the subjects were undergoing. The true human subjects were not stimulated with electrical currents as our digital brain twins, but they had to undergo the progressive matrices task, where there's a series of patterns, and the logical rule for the series needs to be identified, and this last empty box needs to be completed. And the test consists of 24 tasks with increasing degree of difficulty. The first task is easy, the last task is the most difficult one. So this would be a difficult one. And now we look at the performance metrics of the real individuals, not the digital brain twins. And what we see here is the reaction time for the first very simple of the 24 tasks in relation to the general factor of intelligence. And we see that those with a high general factor of intelligence had faster reaction times for the simple, for the first task of the 24 tasks of the fluid intelligence test. So now we look at the last task of the 24 tasks. So that's the most difficult one. And we see that for the last most difficult one, the correlation is inverted. So those with the highest general factor of intelligence took the longest time to take a decision. And we now can look at all the individual 24 tasks. These are the group averages. These are individual subject results. What you see here is the correlation between the score, either the general factor of intelligence or the fluid intelligence score, the progressive matrices task, PMAT, and they behave very similarly. So it's a correlation between the reaction time and either general factor of intelligence or fluid intelligence. And the x-axis shows the 24 tasks. We see that for the first 8 tasks, which are more simple ones, there's a negative correlation indicating with higher fluid intelligence or general factor of intelligence, there are faster reactions. And then with the 9th task, suddenly this inverts. And then for the more difficult remaining tasks, the higher the fluid intelligence was, or the general factor of intelligence, the longer was the reaction time. So in other words, for simple tasks, those with higher intelligence scores were faster. For more difficult tasks, those with higher intelligence scores were slower. And this was predicted also by our model. And we just found this in the empirical data through our predictions of the model. So now we have the mathematical descriptions of our different brains, and we were interested in why are there different performances? Why does the accuracy of the decision-making vary in our 650 virtual brain twins? So what we found was that those with more synchronization in their network, they were the better performers, and those with lower synchronization in the network were the worst performers. And we categorized now these 3 extremes, low network synchrony, high network synchrony, and we also defined an intermediate state and looked what was different in terms of the phase space characteristics in these individuals. So here you see again the simulations, and we stop now the simulation at 0.5 seconds, 1,000 simulations per category, and we look at the phase space at the same time. The phase spaces are reconfiguring during the simulation. This is the status at 0.5 second, and we see that at 0.5 seconds for 1,000 trajectories shown here in red and green, colored according to the decision made either for A or for B. So the axes here are the activity in population A and the activity in population B. And you see here the flow fields. These are the arrows that lead to attractors or fixed points that are indicated as these little balls. So we see that when there's low synchronization in the brain network already after 0.5 seconds, almost the attractors, the decisions for either population B, so when I take it all, population B wins, or population A are taken. If we look at the high synchronized network, we see that the activity between population A and B is still balanced. Yeah? The trajectories here, the red and green lines, are still in a balanced state in the midline. Neither activity A nor activity B is higher. And we also see actually that in the highly synchronized network, we have here in the diagonal, in the balanced state, another fixed point, which we do not see here. And it's exactly this fixed point that keeps the activity in this balanced state. So now we continue the simulation. And we see that after 1 second, all the decisions have been made in the low synchronized network. They either reached the attractor B or attractor A. Also in the medium synchronized network, now after 1 second, most of the decisions have been made in our simulations. And for the high synchronized network, we see that there's still the fixed point, but a little bit moved. And now the trajectories start moving to either A or B decisions. Continue now the simulation. And now after almost 1.5 seconds, we see that also for the high synchronized network, the decisions have been made and the trajectories reached either B or A. And we also see that now the fixed point here that keeps the system in a balanced state has been removed. So we clearly understand how the reconfiguration or the configuration of the phase space as a function of the synchrony between the frontal and the prefrontal regions, where this circuit model is embedded, reconfigures the space. And under low synchronization, there's no fixed point that keeps the two systems in a balanced state. With high synchronization, we have a fixed point here that for longer time holds the two competing populations in a balanced state, which allows for more accumulation of evidence and more canceling of noise, which then, as you can see here, leads to more accurate decisions and less incorrect decisions. So here we have 959 correct decisions and 41 incorrect decisions. For the low synchronized network, we have 790 correct decisions and 210 incorrect decisions. This is a summary. So here you see again how functional connectivity, synchronization changes the input that drives a population. And this is directly related to the percentage of correct decisions, but also the time it takes until a decision is being made. And this is a function of this reconfiguration of the phase space. So we now can mathematically completely understand why there's a variance in decision-making quality or accuracy, and also reaction times. So if we can characterize the brain so well, then of course the consequence is that we want to use these digital brain twins to inform clinical decision-making. And there are clinical studies already using the virtual brain, for example, in the field of epilepsy in France, where the information of the predictions is used to inform the neurosurgeons where to cut the brain in order to stop the epilepsy in these patients. What you see here is another application that we are developing in Berlin, where we have an electrode inserted in the brain digital twin. And this is a multi-scale twin where we have not only the information from the individual patient coming from non-invasive magnetic resonance imaging and maybe intracortical recordings, but on top, we have integrated the information coming from post-mortem digital atlases with a cellular and even subcellular resolution. Here you see the deep nuclei, the basal ganglia, where the electrode is inserted for Parkinson patients to stimulate the deep nuclei and to improve motor dysfunction. And we are presently just in preparation of a pilot trial, clinical trial, to show evidence for the outperformance of this in silico stimulation compared to manual optimization by clinicians. So this is a team in Berlin, and we are very interdisciplinary. So our team comes from, I think, now 26 different countries. There are all kinds of backgrounds that contribute to these simulations. Of course, in order to really now build clinical applications out of our digital brain twins, we need to go through validation certification. And this is not only the case for our digital brain twins, but for all kinds of AI applications. And Europe. I am leading the European Testing Experimentation Facility for Health, AI and Robotics, TEF Health. And I want at the end of the presentation quickly show you this new facility that has started at the beginning of 2023 that has a purpose to ensure that such technologies as digital brain twins are trustworthy. And this facility can be found under tefhealth.eu, provides a single entry point for multiple services that provide technical and scientific support for testing and validation of such technologies. You will see that if you go to this website, you find a knowledge graph. So there's a button at the top, search for services, and this knowledge graph has a multitude of services that are being offered, in particular to SMEs, startups, to the private sector to validate their technologies. And the services provided include secure infrastructure, trustworthy infrastructure, where all these health data can be exchanged in Europe. This is done in collaboration and alignment with European Health Data Space Regulation. Just quickly want to show you the trusted virtual research environments that we are developing with large amount of capabilities, including high performance computing. And these are infrastructures that are fully open source to guarantee also sovereignty and to avoid vendor locking or dependencies from specific vendors. And these trusted virtual research environment are deployed at different IT centers, typically IT centers with high information security levels to ensure the data privacy and security. I'll jump a little bit quicker across these slides. This is just showing that these infrastructures undergo certification, such as Euro privacy certification, to really show their trustworthiness. And here you see a nice resource of eBrains. If you go to eBrains, you will find the knowledge graph there, where the data sets, the models, the software, much of the software I have shown to you can be discovered via knowledge graph search and can be reused. And this is a website of the testing experimentation facility that I just mentioned, where you find the professional services for certification validation of such technologies. And with this, I thank you for your attendance and for your interest, and I would be available for questions.
Thank you so much, Dr. Ritter. This was an amazing talk. And I guess it's a really new topic for most of us here, because we hadn't heard about making a digital model of the brain using different neuroimaging techniques, like how you've done with the human brain twins. And I have a few questions as well. I think there are some in the chat. So my questions, my work is mostly with Parkinson's disease, and we sort of look at the progression of Lewy bodies, or the protein alpha-synuclein, which aggregates between the body and the brain. And I was wondering if we think that there are two kinds of cohorts of Parkinson's, a brain first, brain predominant kind, and a body predominant kind. So could we use this technology of brain twins to sort of find, if we catch an early case of Parkinson's, can we predict which regions of the brain the pathology would then affect in what order, based on all the clinical symptoms and the brain scans and the fMRI scans, PET scans?
Yeah. Thank you very much for this great question. So I think that there's the clear potential. So what we are much actually lobbying for is that non-invasive magnetic resonance imaging of the brain, but maybe even of the entire body, is used on a regular basis throughout Europe. Yeah, we are in Europe, so therefore we are thinking Europe, but to generate exactly this long-term data, longitudinal data, that is required in order to identify early biomarkers and early deviations of the trajectories of different kinds of activities or other processes in the brain and also outside of the brain. Yeah, and this, of course, if we have such early biomarkers, it's the same for Alzheimer's disease. There we are searching actively also for early biomarkers. Of course, there are the UK Biobank, for example, data, but they also only start in healthy adults. And as you rightly said, such neurodegenerative diseases like Parkinson most likely start decades before the symptoms occur.
Yes.
And therefore, early data are required. And we hope with the trend towards really being more careful with how we use data, how we make data fair and can reuse them, and really recognizing the value of such data, that soon such cohorts do exist where also younger human brains can be used to determine whether there are already early changes that can be used as biomarkers. And as we know in Alzheimer's disease, for example, just a few days ago, a new drug has been accepted now in Europe also. It was accepted already in the US, and we were celebrating this actually because we think this is a breakthrough. And we hope, of course, the same will be possible soon in the field of Parkinson's disease.
I hope so. Thank you. So we have a question in the chat. It says, do you think there is a future where all the intracortical recordings from different labs can be somehow integrated to somehow build a functional connectome of human brains for at least some interesting brain parts, like the hippocampus? And in this way, we might look at the average brain activity accounting for all inter-individual differences.
Yeah, this is a great idea, and I believe in principle it could be possible. Yeah, so I do not see what should prevent us. There are, of course, strict regulations in Europe, but even with these regulations, it is feasible. And there are examples already of such multi-center databases, also of intracranial recordings of EEG in epilepsy. Actually, there's this human intracranial EEG platform also being part of eBrains and the Human Brain Project. They already have started exactly such a collection. So I believe it is possible, and it is much needed and would be a very useful resource.
I agree. And the next question is, does the digital brain include data sets from different modalities like neuroimaging and electrophysiology recordings? If yes, can they be somehow trained with multimodal AI models and provide some novel insights into the brain? Do you think there is a future there?
Yes, perfect question. And the clear answer is yes to both questions, actually. So first, there are already studies also from our team. So my background is actually in multimodal imaging, and we do simultaneous EEG, fMRI, for instance. But also, if this is not simultaneously recorded, it can be integrated. We also integrate with PET data, positron emission tomography data. So it's possible to integrate many different modalities in the virtual brain. And do you think there's a future? Yes. And we also actually started combining with AI for different purposes. And there's a perfect future of combining the two technologies and utilizing AI on the one hand to identify certain features in our brain models that can be helpful. But on the other hand, we also believe that our brain network models, for instance, the one I have shown in the second study today, can inform AI algorithms. So we identify new motives, new structural motives that can be also employed in AI to come to even better decisions with AI.
Then we have another one saying, could you expand more on the mathematical correlation of synaptic synchrony and fast or slow problem decision making?
Yeah, this is a big question, but I can refer, if you're interested, to our publication in Nature Communications. I think this is recorded, so you will see also the paper reference, and there all the details are being presented. It's a very complex study, but in a nutshell, as mentioned before, we found that if there's more synchrony in the brain, then this alters decision-making accuracy and also the speed of decision-making. And why that is, we were able to infer using this space analysis, where we very clearly could infer the principles that underlie the different latencies of decision-making and also the accuracy of decision-making.
Right. Yes, and I think for more details, you can find the link to the paper when we upload the recording on YouTube.
Exactly.
And lastly, so Ishan Sheikh says EEG promises a good temporal resolution and fMRI promises a good spatial resolution. For doing both EEG plus fMRI together, do we have to compromise a little bit on both spatial and temporal resolution?
Well, so simultaneous EEG fMRI is very challenging because of the MRI gradients that generate artifacts in the EEG, which are static artifacts. They can be in principle removed, but as soon as the EEG is moving in the field of the scanner, then the static artifacts also are changing. And there's, of course, always a little bit body movement, and therefore this is a challenge to remove these artifacts. But in principle, the combination works very well. If it goes to higher fields like 7 Tesla, then of course you also have to take into consideration potential heating of electrodes, EEG electrodes, and this can prevent actually the simultaneous measurement. And so far, you're totally right. We might have to find the right balance and cannot use the full potential of the EEG or the fMRI as we could use if we would use the modalities alone. But we can also do some measurements separately, and it is also possible, depending how you design your study, to use independently recorded EEG and fMRI signals and to integrate them in a single brain network model.
Right. Yes. Thank you for that. Okay, we have one more. We're getting a lot of questions now, but I think maybe they can reach out to you by email if they have any more questions in the interest of time. And thank you so much, everyone, for joining. And thank you, Dr. Ritter, for coming and giving this amazing talk and enlightening us about this very new topic that I'm very sure is going to have a lot of impact in neuroscience in the future.
Thank you very much again for having me and for organizing this nice event. And I wish everyone, and especially you, success with this event and a lot of fun. So enjoy this great weekend. Bye-bye.
Thank you so much. Bye-bye. And for the other attendants, we have the next session at in 4 hours from now. I say that because I'm sure everyone is from different time zones. So 11 a.m. Eastern time, if that helps. If you're looking for a graduate, if you're applying to graduate school in neuroscience and you want to know how to navigate the process, then we have a graduate school panel in 4 hours from now. So please tune in then. And thank you for joining.